It is actually pretty wild when you stop and think about it. How exactly does a massive oak tree, or even just a tiny blade of grass, manage to grow and thrive? Animals have to constantly hunt or forage for their meals, but plants? They somehow manage to build entirely new physical structures using literally nothing more than invisible gases from the air, a bit of water from the ground, and sunlight. It sounds like straight-up magic, but it's actually one of the most robust and fascinating biological engines on the entire planet.
Welcome to This Explainer. I am absolutely thrilled you're tuning in. Today, we're taking an investigative journey into the ultimate natural manufacturing factory. We are going to break down exactly how plants pull off this incredible feat, acting as these miniature chemical plants that fuel nearly all life on Earth. Okay, here's the roadmap for our explainer today. First, the photosynthesis recipe. Second, using and storing plant food. Third, putting plants to the test. Fourth, measuring reaction rates. And finally, number 5, the limits of photosynthesis. Let's dive right into Section 1, the photosynthesis recipe, the factory that builds life. So, at its core, photosynthesis is simply the process by which plants synthesize carbohydrates from raw materials using energy from light. And the critical machinery making all this happen? It's a green pigment called chlorophyll, which is safely housed inside the chloroplasts of the plant's leaf cells. You can think of this chlorophyll as the ultimate, highly efficient solar panel. It transfers light energy directly into chemical energy. Now, you might be wondering, how do those raw materials actually arrive at the factory floor? Well, the plant ingeniously sources its elements from 2 entirely different environments. First, carbon dioxide diffuses straight out of the air and right into the leaf through these tiny microscopic pores called stomata. Meanwhile, water is being absorbed by the roots deep down in the soil, and it's transported all the way up to the leaves through specialized vessels called the xylem. And that brings us to the actual equation, which really just illustrates the sheer mathematical beauty of this whole process. 6 molecules of carbon dioxide and 6 molecules of water go into the factory. The chlorophyll absorbs the light energy, uses it to literally split those water molecules apart, and then recombines a hydrogen with the carbon dioxide. The end result? Exactly 1 molecule of glucose, that's C6H12O6, plus 6 molecules of oxygen, which conveniently diffuses right back out into the air for us to breathe.
Moving right along to Section 2, using and storing plant food. What happens to the glucose?
Once that glucose is manufactured, the plant has a whole internal economy to run. It's incredibly versatile. The glucose is often quickly converted into sucrose, which acts as a transport molecule sent to areas that don't photosynthesize, like the roots or growing fruits. If the energy isn't needed right away, it gets stored as starch in tubers or within the chloroplasts themselves. It can also be woven into a tough fibrous armor called cellulose to build cell walls. Sometimes it's mixed into sweet nectar to attract insects for pollination, or even combined with nitrogen from the soil to build vital amino acids and proteins. It's the ultimate building block. All right, Section 3, putting plants to the test, proving the laws of photosynthesis. Now, it's one thing to talk about this factory, but how do we actually prove it's running? We look for our smoking gun, starch. Since starch is made from the glucose the plant produces, its presence proves photosynthesis happened. We test for this using the iodine starch test. It's basically a 4-step recipe. First, we boil a leaf in water for about 30 seconds to break down the cells. Second, we submerge it in boiling ethanol to strip away all that green chlorophyll so we can actually see the color changes. Third, we soften the brittle leaf in some hot water. And finally, we apply an iodine solution. If starch is present, that iodine instantly transforms from a rusty brown color to a deep, dark blue-black. We can use this exact test to prove that light is absolutely non-negotiable for this process. Imagine taking a leaf that's still attached to a plant and wrapping a portion of it tightly in foil so it gets 0 light, leaving the rest completely exposed. What happens after a few days? Well, when we run our iodine test, the exposed parts turn blue-black. They're just bursting with starch. But that covered part under the foil, it remains totally brown. No light means no glucose, which means no starch. We can also run a really clever isolation test for carbon dioxide. Picture 2 plants, each sealed inside a clear bag. Plant A is trapped in there with a dish of soda lime, which is a chemical that aggressively absorbs all the carbon dioxide out of the air. Plant B is trapped with sodium bicarbonate, which actively releases extra carbon dioxide. After leaving them in the light for a few hours, we test the leaves. Plant A's leaf stays brown in our iodine test. Starving it of carbon dioxide completely shut the factory down, while Plant B thrived.
And our biological detective work doesn't stop there. We can prove chlorophyll is required by testing variegated leaves. You know, the ones with those white patches? Only the green parts of those leaves will turn blue-black with iodine. Plus, we can even prove oxygen is being made. If we submerge an aquatic plant under a funnel, we can actually trap the invisible gas it releases in a test tube. If you take a recently blown-out glowing splint and stick it into that tube, the concentrated oxygen will literally reignite it into a flame.
Section 4. Measuring reaction rates. Speeding up the factory. So we know photosynthesis happens, but scientists also want to know how fast it's happening. To track this dynamic shift, they use an ingenious visual alarm system called the hydrogen carbonate indicator. If you place a piece of pondweed in this liquid and shine a light on it, the solution turns purple. That tells us carbon dioxide is rapidly being sucked up by photosynthesis. But if you stick that exact same setup in a dark closet, the solution turns yellow. Without light, the plant stops photosynthesizing and is actually just respirating, releasing carbon dioxide back into the water, exactly like we do when we exhale. To actively measure the actual speed of the reaction, we can just sit back and count the oxygen bubbles that an aquatic plant releases in 1 minute. It's remarkably simple but effective. As you can see, when a light source is placed just 10 centimeters away, the rate of oxygen bubbles is really high. But when we slide that same lamp back to 20 centimeters, the bubble rate drops off significantly. This beautifully illustrates that the intensity of the light directly dictates the speed of the whole photosynthetic reaction.
Finally, Section 5, the limits of photosynthesis, hitting the ceiling.
Okay, so what happens when we try to push this plant factory to its absolute maximum capacity? We hit a wall, known in biology as a limiting factor. A limiting factor is simply something present in the environment that restricts a life process because it's in short supply. Basically, your factory's total output is always going to be limited by whatever its rarest resource happens to be. For photosynthesis, it usually comes down to a triad of external limiting factors: light intensity, temperature, and carbon dioxide concentration. Here's the catch. If you give a plant endless, blinding sunlight, its production is still going to plateau if it runs out of carbon dioxide. Similarly, because this whole process relies heavily on enzymes, if the temperature drops and it gets too cold, those enzymes slow way down. The whole factory stalls out, even if it's a brilliantly sunny day. Just think of a small fern on a deeply shaded forest floor. Even if it's incredibly warm and there's plenty of CO2, the lack of light punching through the canopy acts as its limiting factor. Now, connecting this micro-level biology to macro-level agriculture is where things get truly exciting. This isn't just stuffy textbook theory, it is the literal foundation of modern farming. Farmers actively combat these natural limiting factors all the time using polytunnels and massive greenhouses. Inside these tightly controlled environments, they can artificially jack up the temperature, turn on massive grow lights, and even pump in extra carbon dioxide, pushing their crop yields to the absolute maximum potential to feed a very hungry world. Which leaves us with a pretty massive, provocative thought to wrap up on. As our global population continues to skyrocket and farmable land becomes harder and harder to find, how might manipulating these exact limiting factors in high-tech greenhouses be the ultimate key to solving future global food shortages? By perfectly optimizing this ancient, invisible plant recipe, we just might hold the power to sustain the future of humanity. Thank you so much for joining me on this explainer. Keep asking the big questions, and I'll catch you next time.